The Ebola virus is part of the viral family Filoviridae, which also contains the Marburg virus and Cueva virus. The first outbreaks of Ebola occurred in the villages of Africa, with the most recent outbreak occurring between 2014 to 2016. It has a vitality rate of 50 to 90% of cases. It is believed it was first transferred from bats. One can get Ebola by being in close contact with infected animals such as bats, gorillas, monkeys, or chimpanzees. Ebola has negative stranded RNA, which means it has a single-stranded RNA genome that is opposite of the coding strand. It is also non-segmented, meaning the RNA is on one strand rather than in multiple smaller strands. The Ebola virus contains 7 genes, making up 8 total proteins. The structure of the Ebola virus contains a long filamentous viral envelope. The outside is covered with glycoproteins that help it enter the host cell. Inside the virus, there is a protein nucleocapsid that wraps snugly around the RNA. The polymerase protein is responsible for its replication once it enters into the host cell. The other essential part of this virus is VP40, or viral matrix protein 40. VP40 protein is a 40 kilodalton matrix protein. The matrix is in the layer between the outer envelope and the nucleocapsid. It gives the protein its shape. The structure of VP40 changes depending on its responsibility in the cell. It can exist in 3 forms: dimer, hexamer, or octamer. Through a series of mutagenesis and crystallization experiments, the functions of each of the conformations was determined. The dimer is a homodimer with 297 residues. It dimerizes in solution. When the dimer packs as a crystal, it organizes itself with the NN and CC domains lined up with each other. To determine the function of the dimer, the NN interfaces were mutated. Threonine 112 and leucine 117 were the essential residues for the NN terminal interaction. Both the L117R and the T112R mutations inhibited the protein's ability to migrate to the membrane and bud viral particles. Mutating the NN interface disrupted the viral budding. The CC interface was also mutated. Methionine 241 and isoleucine 307 are important for this interaction. M241R did not bud or migrate well, and the I307R mutation did not migrate or bud at all. Therefore, the dimer plays an essential role in membrane trafficking and budding of virus-like particles. It was thought that the dimer C-N-N-C interaction was how the matrix was assembled. However, the dimer model was not consistent with 2 known facts. First, the oligomerization into hexamers when it interacted with the membrane, and 2, the NC terminal domains separated from another. To figure out how the protein binds the membrane, scientists did a crystallization experiment. They used dextran sulfate as a substitute ligand. VP40 normally binds phosphatidylserine in the membrane. Dextran sulfate was used to mimic the ligand-protein interaction. The crystallization experiments showed that binding to the ligand triggered a conformational change into a hexamer. This configuration is consistent with the NC terminal domains separating and the hexameric oligomerization. This conformation is also consistent with the matrix structure of Ebola. The Ebola matrix has 3 layers, an outer, a core, and an inner layer. The hexameric structure lines up with the data present, and it was determined that the hexamer is important for the matrix assembly. For the octamer, the residual arginine 134 was known to be important for the RNA binding and ring assembly of VP40 in the octameric form. This residue was mutated to an alanine. The result of this mutation showed that the virus can still bud and assemble the matrix, but it is actually a lethal mutation. How is this possible? VP40 has yet another function outside of assembling the matrix. It regulates viral transcription in the infected cells. When this residue is mutated, it can no longer control the transcription of the RNA. How the protein changes conformation between the dimer, hexamer, and octamer is not completely clear, but possible mechanisms have been proposed. From the dimer to the octamer, it is believed the NC terminal domains break apart, then the NN interface breaks apart, and then the monomers then rotate 180 degrees and backwards. 4 dimers in this configuration assemble and form an octamer. An RNA binding domain is now made that was not there in the dimer configuration. For the dimer to the hexamer, there is a filament intermediate with dimers linked in the NN and CC conformation. When this filament reaches the membrane, a conformational change occurs that flips the middle dimer. This further causes the C-terminal domains to flip in and out of the filament. In this conformation, the 2 dimers on the ends are in the regular NC configuration as seen in the dimer. The 4 in the middle have an NN interaction with the C domains pointing out. The hexamers come together via CC interface interactions that occur in the dimer. There is an important tryptophan residue that links the 4 inner domains together that is exposed when the C-terminal is released. In conclusion, VP40 serves many functions for the virus. In the dimer form, VP40 migrates to the membrane and buds viral-like particles. Once it binds to the membrane, it changes conformation to a hexamer that assembles the matrix. And once it infects the host cell, it forms an octameric ring that aids in viral transcription. Although there is no current vaccine for the Ebola virus, understanding the role of VP40 may lead to possible targets in the future.